deoxyribonucleic acid, DNA. Why are scientists and doctors so interested in its wonders? Well, DNA is like an encyclopedia. The words inside are composed of only 4 letters: A, adenine, G, guanine, C, cytosine, and T, thymine. When scientists examine DNA, they carefully read each and every word inside this complicated encyclopedia. When the words finally begin to make sense, marvelous things can start to happen. But how do we retrieve these words, or DNA sequences? Analyzing DNA sequences have aided in several scientific fields since the 1900s, from Sanger sequencing to applied biosystems to the genome analyzer. Scientists clearly have an insatiable hunger for finding the next best technology to accurately sequence large portions of DNA. Next-generation sequencing, also known as NGS, has fundamentally changed the scientific world. It is important to note that the costs regarding next-generation sequencing are significantly lower than in any other point in DNA sequencing history. Illumina is a largely successful biotechnology company that plays a large role in next-generation sequencing. They have 4 steps that briefly explain the complicated process regarding this version of DNA sequencing: library preparation, cluster generation, sequencing, and data analysis. Let's start from the beginning. The first phase of this process is library preparation. Essentially, a DNA sample is broken down, and specialized adapters are glued to the 2 sides of the fragmented DNA. These 2 steps are done simultaneously, which helps immensely with the proficiency of this stage. The new DNA fragments are then led to polymerase chain reaction, or PCR, amplification. They are then gel purified. The second phase is cluster generation. A DNA library is carefully placed into a flow cell. The DNA fragments attach to the top of the flow cell. This is a complicated step, where the flow cell contains oligos that complement the incoming library adapters attached to the DNA fragments. This step is not complete until every DNA fragment goes through a bridge amplification cycle. The second phase is tedious, but it is extremely important for entry into the third phase. The third phase is sequencing. Nucleotides are added, and Illumina technology is able to identify single bases as they combine into each DNA strand. The nucleotides in this step are very special because they are fluorescently labeled. This helps the scientist carefully analyze each cluster, where the first bases within each of the 4 clusters can be visualized due to fluorescent lights. The final phase is data analysis. Once the scientists are able to visualize the various DNA sequences, they are sequentially aligned using a reference DNA sequence, also known as a reference genome. A reference genome is a man-made DNA sequence that expresses a specific species set of genes. This phase can only be done using an advanced bioinformatics program. This program helps distinguish any variances between the align reads and the reference DNA sequence. Computational methods are then used to aid in the creation of visual models, such as phylogenetic trees. Overall, next-generation sequencing has become a popular process that can aid in various scientific fields. Millions of DNA sequences can quickly be processed at astonishingly low rates. This is only the beginning of a new scientific era.